evening, everyone. I would like to welcome all of you for this session today on a journey through images for a quick revision in embryology on systematic embryology. This is the third part of the entire series which we have run across the period of last 1 week. This is the third part of it. We again welcome you all. I will hand over the talk to Priyanka to introduce the author, and then we'll begin with the session here.
Thank you, Kritika. I would like to welcome everyone, all the participants here in this third masterclass of our series, where we are here to revise embryology quickly with Dr. Rose Xaviour. Let me first start by giving a brief introduction of our speaker, Dr. Rose. She is an assistant professor in the Department of Anatomy at Government Medical College, Thrissur, Kerala. She completed her graduation and post-graduation also from the same institute. She has published more than 10 national and international scientific articles in various journals, and she is an assistant editor of the National Journal of Clinical Anatomy. Recently, Dr. Rose has been selected by NPTEL Swayamprabha Academic Channel under Ministry of Human Resource Development, Government of India, to deliver video lectures on embryology. Welcome, Dr. Rose.
Thank you.
Dr. Rose has already discussed Dr. Rose has already discussed general embryology and systemic embryology in the previous two masterclass sessions. So today she will be covering the topic of central nervous system and eye. But as promised, Dr. Rose, we will begin by answering some of the questions asked by the participants in the last session. So we have identified 4 questions, Dr. Rose, which were asked by multiple participants. So should I read them to you now? Yeah. Okay. So the first question, Dr. Rose, is, can primary oocytes arrested in diplotin stage of prophase 1 can be a reason for the increase in birth defects among older mothers?
Actually, the thing is, when a female baby is born, the baby is having the oocytes meant for its reproductive period. So whenever a cell is actually remaining for a longer period, there will be DNA damage, and that is the proteins which bind the chromosomes will be damaged over years. And in this particular question, I think the person has asked about the trisomy, the anomaly called trisomy. So what happens is, again, during cell division, the chromosomes won't be actually getting separated. So they will remain as a pair in case of oocyte. And it will actually, during fertilization, what happens is one single chromosome will come according to the set of chromosomes. If it is for the 21st, we have Down syndrome, like that. So ultimately what happens is, instead of a single pair, we have 3 chromosomes in one set of number. So that is the reason why you call it as non-disjunction of chromosome. So that is the reason why we get more cases of Down syndrome in ladies who get pregnant towards older age, say roughly after 35 years of age. Priyanka, one more thing, I think the slide is not moving.
Okay, Dr. Rose, can you just try again, or I will upload the presentation at my end then.
Okay, I'm trying. Yes, okay. Any more questions? Hello, Priyanka, can you hear me?
Yes, Dr. Rose. So I will read the next question to you. The next question is, what is the fate of primitive streak?
Actually, the primitive streak, when it's formed in the epiblast, it gives rise to all the 3 germ layers, like the endoderm, the mesoderm, and the ectoderm proper. So it's usually formed during the 15th day. And later what happens is, after 4th week, it usually start regressing, or it will start diminishing in size, and finally it just disappears. Sometimes the primitive streak will stay there at the caudal region. And you know, the primitive streak is made up of primordial germ cells, isn't it? And they are, it's made up of pluripotent cells, so that they can give rise to any type of cell. So what happens is, if this primitive streak is seen as a remnant in the caudal region, this will give rise to a tumor which will contain derivatives of all the 3 germ layers. Okay, and since it is seen in the caudal region or in the sacral region, you call this tumor by the name sacrococcygeal teratoma. The word meaning of teratoma is it will be made up of derivatives of all the three germ layers. So that is the fate of primitive streak. Usually, when the baby is born, there won't be any remnant of primitive streak. If at all present, since it is made up of pluripotent cells, it will give rise to derivatives of all the three germ layers, and you call it as teratoma. Yes.
Okay, thank you, Dr. Rose. So the next question I have is, during IVF, is the nucleus of the sperm injected or the whole sperm is placed over ovum?
That's a very interesting question. Actually, during in vitro fertilization, what happens is we are getting the sperm and ova from the donors, and we are creating an artificial environment just like the natural one, so that the sperm and ova will get fertilized as they do in vivo. For this, we actually require properly functioning sperm number, isn't it? To complete the sequence of events leading to fertilization. But in severe cases of male factor infertility due to lack of sperms in ejaculation, like in cases of severely impaired spermatogenesis, or in some cases where there is non-reconstructible reproductive tract obstruction, in those cases what we have, the condition is we have only a limited number of sperm, so we can't take the risk of just leaving the sperm with the ova. So what we have to do is we have to do some manipulations, you call it as micro manipulation, and some of the steps we use are, in some cases we have to keep the sperm just under the zona pellucida, you call it as subzonal sperm insemination. Sometimes you have to make a nick over the zona pellucida, you call it as partial zona dissection, and sometimes we even have to keep the sperm inside the cytoplasm of the oocyte, you call it as intracytoplasmic sperm injection, otherwise known as ICSI. So that's it. So it's not always leaving the sperm with the ova. Sometimes we have to go for some micro manipulations as well.
Thank you, Dr. Rose. So just one more question before we start today's session. And this question is, what is meant by Meckel's diverticulum?
So when we mentioned about the folding of the embryo, the cephalic folding, the caudal folding, the lateral folding and all, the formation of primitive gut, we mentioned about a duct known as vitalointestinal duct, which connects the midgut with the yolk sac. Sometimes this duct will persist, and this persisting duct is known as Meckel's diverticulum. And there is a very interesting rule of 2 followed by Meckel's diverticulum. That is, it is seen in 2% of population. It presents before 2 years of age. It is 2 inches long. It is 2 feet away from the ileocecal valve, and it contains 2 types of heterotopic mucosa, like it contains the pancreatic and gastric mucosa. So this is an interesting rule of 2 followed by Meckel's diverticulum.
Okay, so Dr. Rose, now before we start the session, just two or three points I need to tell to the audience. Number one, audience, we are recording this session, and a recorded version will be shared with you after the session. So all the participants will be able to get a copy on email afterwards. Secondly, I want to say that there is a Q&A tab in your screen. So whatever queries you have, you can post your queries in the Q&A tab, and Dr. Rose will answer to them after the session today. And one more thing I need to tell everyone is that there will be some polling questions in between the sessions. So the answers are anonymous. So please feel free to answer them and keep this session going live. Thank you. So Dr. Rose, you can continue with the session now.
Thank you so much. So we are moving on to today's session. It is systemic embryology, and we'll try to cover the most important topics which are always asked, that is development of central nervous system and something about the development of eye. So under the development of central nervous system, you have to be familiar with these terminologies, like what do you mean by neurulation, what do you mean by the neuropause, their closure, then fate of neural crest cells, fate of neural tube, development of brain in the form of rhombencephalon, mesencephalon, prosencephalon, and something about the development of suprarenal gland and pituitary gland. So let's have the first polling session. Primary inducer of neurulation. Which among the options you feel correct?
have 5 more seconds left.
Okay.
Here are the results.
Okay, so I think we'll come to it. So thank you. You can close the poll. So during embryonic development, what happens is whenever one structure is formed, it will induce the formation of the other. So that is what is meant by inducer. And as I think some of you got confused with primitive streak. Primitive streak is considered as primary organizer. So neurulation is the process by which neural tube is formed, and neural tube is formed from the neuroectodermal layer. That is what is meant by neurulation. So when you look at this diagram, you can see a dot-like thing, the black dot. That is the notochord. And by the formation of notochord, we have the surface ectoderm. The entire thing was ectoderm. By the formation of notochord, the region of the ectoderm lying over it will get differentiated to form neuroectoderm. Okay, so in this condition, the question was which is the inducer for neurulation. So in this condition, the notochord is acting as the inducer for the formation of neural tube. So we know that the ectoderm is actually getting differentiated into neuroectoderm, which is lying over the notochord, and the remaining part will be known as surface ectoderm. So this neuroectoderm will first form a plate known as neural plate. Then what happens is, you know, on either side of the notochord, we have the mesoderm, the intraembryonic mesoderm. As the cells are actually getting replenished here, there will be a projection on either side of the notochord upwards. So what happens is there will be a depression in the neural plate, and this depression is known as neural groove. And the junction of the neural plate with the ectoderm, with the surface ectoderm, this junction, you call it as neural fold. So at this point of time, we have a neural groove formed from the neural plate, and you have two neural folds on either side. That just means the junction of the ectoderm, the surface ectoderm and neuroectoderm. Now what happens is, when you consider the entire embryonic disc, from the cervical region, these folds will start getting approximated in the midline. So it will start in the cervical region and it will go cranially as well as caudally. Okay, so for the time being, we know that there are two openings at either ends of the neural tube. So you know what is the meaning of keeping these two openings patent? Because during the development or neurulation, we don't have a very established circulatory system to provide nutrition for the developing neural tube. So what happens is, we have the amniotic cavity above, isn't it? This is the ectoderm and we have the amniotic cavity above. So if this tube is kept open at both ends, what happens is the amniotic cavity can run through the tube and it can nourish the developing neural tube. So that is the reason why the tube is actually kept open at both ends. So it starts at the cervical region, and the anterior opening, you call it as anterior neuropore or cranial neuropore, and the posterior end you call it as caudal neuropore or posterior neuropore. And finally what happens is this will get approximated in both directions and it will close. Both the anterior neuropore as well as posterior neuropore will be completely closed, so that the tube will be a single closed tube. So the anterior neuropore usually closes by around 20 somoid period, and the posterior neuropore closes by around 25 somoid period. So when they close together, you can see that the neural fold cells are not actually getting incorporated into the neural tube. They are actually lying as a separate mass just above the neural tube in the midline. And these cells are actually known as neural crest cells. So in the beginning, the neural crest cells will be a single mass which is lying on the dorsal aspect of the neural tube. Later what happens is it will just split into two and it will just lie on either side of the neural tube. And further what happens is this neural crest on either side will again divide into a dorsal mass and a ventral mass, and they will just migrate dorsally and ventrally. So the neural fold cells, soon after closure, they are not getting incorporated into the neural tube, They are actually forming a separate mass known as neural crest cells. They will be first seen as a midline mass. Later they split into two so that they lie on either side of the neural tube, and further again they split into dorsal and ventral mass, and they will migrate dorsally and ventrally. So so this is actually a fetus which got aborted, say roughly around 28 weeks of gestation. And let's have a polling question now. Which is true about the given clinical condition? Is it failure of closure of anterior neuropore, or the brain fails to develop, or it is diagnosed prenatally and terminated? If 400 micrograms of folic acid daily 3 months before conception and throughout pregnancy given for prevention can prevent this condition, or are you saying all the statements are true? Please give your answers.
5 more seconds to go.
Very good response. I think all of you knew this. So this is a condition known as anencephaly. Anencephaly means the failure of closure of the anterior neuropore. So can you please close the poll? So that is a condition known as anencephaly. We'll come to it later. So for the time being, again, you need to know about the derivatives of the neural crest cells. This is a favorite question for almost all the exams and MCQs as well. So we have mentioned that the neural crest cells are actually dividing into two groups, and they are migrating dorsally as well as ventrally. Roughly speaking, the dorsally, they will form the melanocytes in skin and hair follicles, and ventrally they give rise to the sensory ganglia, sympathetic and enteric neurons, the Schwann cells, and cells of the adrenal medulla. And if you just have a look at this diagram, it will actually give the entire picture of the neural crest cells. So in this region, you can see the cranial neural crest cells. The neural crest cells seen in the cranial region are giving rise to all these derivatives. You can see that it is giving rise to the connective tissue component of the cranial muscles, the thymus, the adenohypophysis, the parathyroids, and the thyroids. Then somewhere here you can see the pupillary and ciliary muscle, then type I cells of the carotid body. Then we have already mentioned about the dermis of the face and skin, then the bronchial arch cartilages, and some contribution to the membrane bones of the skull. When you talk about the neural crest cells seen in the spinal region, this is the limit. Okay, so in the spinal region, you can see that it is giving rise to the sympathetic ganglia, parasympathetic ganglia of neck. Then you have the pre-aortic ganglia, adrenal medulla, very important. Then also you have the dorsal root ganglia and the sympathetic chain. Now, the neural crest cells seen in both regions, cranial as well as the spinal region, will give rise to the truncoconal septum. We have seen the septum which divides the truncus arteriosus into the pulmonary artery and the aorta. Then you have the glial cells, the Schwann cells, the melanocytes, all these enteric ganglia, all these are formed from the neural crest cells seen in the cranial and spinal region. So that's about the derivatives of the neural crest cells. So let's see one more polling question.
The polling is in front of your screen right now.
Okay, so the question is, all are derivatives of neural crest cells except. We have seen many derivatives of neural crest cells right now. Out of the 4 options, I haven't mentioned one option as the derivative of neural crest cells. So
5 more seconds.
very good. So all of you are happy to know that all of you are attending the session very properly. So adrenal cortex. Adrenal cortex is actually mesodermal in origin, whereas the adrenal medulla is actually derived from the neural crest cells. So let's move on.
Please go ahead, Dr. Rose.
Yeah, I think There is some issue with the screen.
Just wait for about a second. I think it'll be fine.
Yeah. Okay.
Are you facing any challenge?
No, it's. I think the screen is actually stuck.
Yeah, so just try to close the screen and upload it again.
Okay, fine. So, sorry for the interruption. Now we are moving on with the development of the central nervous system. So we are in a stage where we have a neural tube formed with both the anterior neuropore as well as the posterior neuropore closed. Now we have a single neural tube and it is actually giving rise to the central nervous system. So at the cranial end, the neural tube is actually forming three important brain vesicles from which we have further development of the central nervous system within the cranial cavity, and the caudal end is actually giving rise to the spinal cord. So the cranial end, we have the three brain vesicles, namely the prosencephalon, which is known as forebrain, mesencephalon, which is known as midbrain, and rhombencephalon, which is known as hindbrain. So we have seen the neural tube formed. This is the anterior neuropore, this is the posterior neuropore. Once these neuropores are closed, we have a single neural tube formed. At the cranial end, this neural tube will be having three brain vesicles. They are known as prosencephalon, mesencephalon, and rhombencephalon. Prosencephalon is otherwise known as forebrain. These subdivisions you should be very thorough with, because all these things are favorite question for the examiners. So prosencephalon is actually giving rise to telencephalon and diencephalon. The two subdivisions of prosencephalon, you call it as telencephalon and diencephalon. And they will be giving rise to the cerebral hemispheres as well as the thalami and the associated structures. Mesencephalon, it is not having any subdivision. It is just giving rise to midbrain. And rhombencephalon, it is actually further divided into metencephalon and myelencephalon. Once again, rhombencephalon is getting divided into metencephalon and myelencephalon. And metencephalon will be forming the pons and cerebellum, and the myelencephalon will be giving rise to medulla. So why you call this as brain vesicle? We know that there is neural tube, and at the cranial end, they are getting dilated and it is filled with fluid. So as these forebrain, midbrain, and hindbrain are getting differentiated, we know that there are some cavities enclosed within these. So let's see what are the cavities enclosed within each part of the brain. So in the cerebral hemispheres, we know that it is the lateral ventricle. Between the thalami, between the diencephalon, we have the third ventricle. Within the midbrain, the cavity is known as aqueduct. Within the pons and cerebellum, you have the upper part of the fourth ventricle, and again with the medulla and cerebellum, we have the lower part of the fourth ventricle. So again, this thing you have to keep in mind, the cavities enclosed within different parts of the brain. So prosencephalon, it is divided into telencephalon and diencephalon. Mesencephalon, there is no subdivision, and rhombencephalon divides into metencephalon and myelencephalon. So let's have a polling question now. Aqueduct of Xaviour is the cavity enclosed in. We just mentioned about the cavities in each part of the brain, right? So aqueduct of Xaviour is seen in which part of the brain, or it is enclosed in which region? That is the question. Very good. So the answer is mesencephalon, because we know that in telencephalon, we have the lateral ventricle, diencephalon, we have the third ventricle, and the third ventricle is connected to the fourth ventricle within the rhombencephalon through a duct which is seen in the midbrain, and the midbrain, another term, or the midbrain is derived from mesencephalon. So the cavity of mesencephalon, you call it as aqueduct of Xaviour. Okay. Now, when the neural tube is actually developing within the cranial cavity, we know that the cranial cavity is a fixed space. Just like when we mentioned about the development of heart tube, we know that within the pericardium, if it just gets elongated, it won't be accommodated within the pericardium. So it is just folding on itself to form the bulboventricular loop. Likewise, the neural tube again, if it enlarges, it has to get accommodated within the cranial cavity. So it is actually undergoing so many flexions. So you call it as flexures. So let's see the important flexures seen here. So first one is telencephalic flexure. So telencephalic flexure is a flexure seen between the telencephalon and diencephalon of the forebrain. Between the telencephalon and diencephalon, we have the telencephalic flexure. Then the next one is mesencephalic flexure. So can you see a flexure here? This is known as mesencephalic flexure, and that is a flexure seen within the mesencephalon. The mesencephalon is folding on itself, and that is known as mesencephalic flexure. Then the next one is pontine flexure. So this region is actually the rhombencephalon, which is further divided into metencephalon and myelencephalon, right? So between the metencephalon and myelencephalon, there is a flexure, and this flexure is known as pontine flexure. At this point, I would like to mention one more thing. Here you can see that the pontine flexure is getting deepened, so that there is a lip formed in the upper part, and this you call it as rhombic lip. So when you are asked to mention about the developmental source of cerebellum, so it is from the rhombic lip. We don't have time to finish all the minor details, so that's the reason why I'm adding this point here. With the formation of pontine flexure from the metencephalon, you have a lip formed that is known as the rhombic lip, and it is the rhombic lip which is giving rise to the formation of cerebellum. And the last flexure, the important flexure which we have to mention here, is known as the cervical flexure, and cervical flexure is actually seen between the spinal cord and the rhombencephalon. That is the cervical flexure. So these are the most important flexures you need to know in the development of central nervous system, because all these again can be asked in various MCQs. Now we'll move on to the next slide. First, we will see the internal architecture of the spinal cord, how the development of spinal cord is happening within the neural tube. So we know that the neural tube, after its formation, it will be lined by a group of cells known as neuroepithelial cells. Okay, so the neuroepithelial cells will then just get converted into a group of cells known as neuroblast. And the neuroblast will be actually forming the mantle layer. Can you see the mantle layer here? The blue colored region, that is the mantle layer. And the fibers arising from this neuroblast will be terminating at the periphery, and they will be forming the marginal layer. So once again, the neural tube will be having a lining epithelium that is known as neuroepithelial cells. They will be actually getting transformed to form neuroblast cells. The neuroblast cells will be arranged towards the cavity, and they are known as they form a layer that is known as the mantle layer, and the nerve fibers arising from the mantle layer will form the outer marginal layer. So we know that the mantle layer is actually giving rise to the gray matter, because in the spinal cord we know that the gray matter is inner and the white matter is outside. So the mantle layer will be giving rise to the gray matter, and the marginal layer will be giving rise to white matter. Now we need to know how are the cells within the mantle layer further arranged. There is a sulcus. You can see a oblique horizontal sulcus separating the two parts of the mantle layer. Dorsally, you call it as, so after the formation of the sulcus limitans, you call the mantle layer dorsally as the alar plate, and ventrally you call it as basal plate. So alar is actually the sensory part, and basal is actually giving rise to the motor neurons. So you have the mantle layer further subdivided into alar plate dorsally and basal plate ventrally, and the basal is motor whereas the alar plate is sensory, and we have the sulcus limitans separating these two plates. And so this is the spinal cord, and as we move up towards the brainstem, what happens is soon after the spinal cord we have the medulla, right? So till now, the spinal cord was just in the form of a tube. And when it reaches the medulla, what happens is it will be opened up just like you open a book, isn't it? So what happens is the dorsal ala plate will be now lying laterally. You can see the dorsal ala plate lying laterally. And of course, the basal plate is almost towards the midline. And the sulcus limitans, the position of sulcus limitans also shifted towards the lateral aspect. So it is not just lying dorsally and ventrally. When it reaches the medulla, it is lying medially and laterally. And you know, the basal plate is actually further getting subdivided into the efferent neurons. We know that the functional columns of the cranial nerve are arranged in this manner. So we know the efferent set of neurons are actually derived from the basal plate, and the efferent set are derived from the ala plate. So from the basal plate, we have the somatic efferent, which is lying more towards the midline. Then we have the special visceral efferent, and followed by the general visceral efferent. If you have a clear-cut knowledge of this formation, it will be easy for you to know the functional column formation of the different cranial nerves. So somatic efferent, what do you mean by somatic efferent? So they are the neurons concerned with the skeletal muscle. Then special visceral efferent means they are going to supply the branchial muscles. Special visceral means always related with branchial arch derivatives. So since it is efferent, it is motor and it will be supplying the branchial arch muscles. General visceral efferent means it will be supplying the smooth muscles and glands. Now talking about the sensory component, we have first the general visceral efferent, which carries general sensations from the viscera. Then the next group is special visceral efferent, especially the taste sensations. Then we have the somatic efferent. Somatic efferent, it can be touch, pain, temperature from different parts. So this is how the dorsal ala plate and ventral basal plate gets shifted when it ascends up in the brainstem. So in the brainstem, the arrangement, the pattern of arrangement is a bit different from that of the spinal cord. So likewise, the regions will differ in pons, in midbrain also. I'm not going to the detail right now. So this is actually a newborn. And you, sorry, can you please close the poll? Priyanka? Okay, thank you. So this is actually a newborn. You can see on the dorsal aspect towards the sacral region, there is a defect. You can see something fleshy, right? It's not covered by skin. So what is this condition? What is the baby suffering from? So let's have the poll. Identify the neural tube defect. So in which clinical condition we will get this picture, or in which defect you will get this clinical condition? Identify the neural tube defect. Anyway, it is a neural tube defect. So what it is? Very good. So that is spina bifida. This condition is known as spina bifida. We have already mentioned that if the anterior neuropore is not closed, you get anencephaly, and depending upon the severity, we have many different types of anterior neuropore closure defects. Similarly, if the caudal neuropore is not closed, we get spina bifida, and again you have different grades of spina bifida. So let's have a look at the what all defects you get if the neural tube is not formed properly. So this concept you have to make it clear, because I have seen students finding it a bit confused. That is the reason why I included this slide. So this figure, we can see that from cranial end to caudal end, the entire neural tube is just exposed to surface. So this condition is known as craniorachiasis, and this is actually said to be a very severe or serious anomaly. Now, these three figures are actually anomalies related to failure of closure of neuropore, the anterior neuropore, the cranial neuropore. If the cranial neuropore is not closing properly, you will get all these three conditions. The first one is anencephaly, where the brain is not at all formed. In the second one, you get meningoencephalocele. Encephalon means brain. So meningoencephalocele means you have the meninges along with the brain protruding through a defect. And this condition is known as meningocele, the third condition. Meningocele means just the meninges with the fluid is herniating. The brain, the encephalon, is not herniating. So these are the three defects which you get when there is a defective formation or defective closure of the anterior neuropode. Now coming to the neural tube defect at the caudal end, we have these three defects. The first one is myelocele. So what do you mean by myelocele? So myelocele means the spinal cord is exposed to outside. Okay, myelo means something related with the spinal cord. So spinal cord is actually exposed to the surface. The next condition is meningomyelocele. Meningomyelo means the meninges as well as the spinal cord is exposed. So here the difference between the myelocele and meningomyelocele is, in meningomyelocele, we have the meninges covering the spinal cord. And the third one is meningocele. Meningocele means only the meninges is actually protruding, whereas the spinal cord is not protruding. So if you understand these terminologies, it will be very easy for you to understand many clinical conditions. So these are the main set of neural tube defects which you will come across. Now we will move on to the development of suprarenal gland. When you talk about suprarenal gland or adrenal gland, you know that there is a cortex which is covering, and there is an inner medulla, adrenal cortex as well as adrenal medulla. So adrenal medulla, this blue colored region, it is actually derived from the neural crest cells. We have already seen it. Now what about the adrenal cortex, the outer part of the adrenal gland? The adrenal cortex is actually developed from the mesoderm just lying around the adrenal medulla. So the first formed mesoderm of the adrenal gland, you call it as fetal cortex. So the first formed mesoderm which is covering the medulla, you call it as fetal cortex. And as the medulla is actually getting incorporated into the fetal cortex, there is another layer of mesoderm formed around the fetal cortex, and that is actually the definitive cortex or the permanent cortex. Now in course of time, what happens is you can see that the fetal cortex is actually diminishing in size, and soon after birth you are not getting any remnant of the fetal cortex. So where are the other layers coming from? So at birth, you know, so there are mainly three layers for the adrenal cortex. From outer to inner aspect, you have zona glomerulosa, then you have zona fasciculata, and the third layer is known as zona reticularis. These are the three layers of adrenal cortex. So soon after birth, you will be having zona glomerulosa and zona fasciculata derived from the definitive cortex, and later on the zona reticularis will be formed to the inner aspect of the cortex, and it will be lying closer to the adrenal medulla. So for the time being, you have to remember two things. Adrenal gland has got cortex, outer cortex and inner medulla. Medulla is from neural crest cells, and the cortex is mesodermal in origin. And the cortex, the mesoderm first forms the fetal cortex, and outer to it you have the definitive cortex. And soon after birth, the fetal cortex will just diminish in size and will just disappear. And the outer cortex, the definitive cortex, will give rise to all the three layers, the zona glomerulosa, fasciculata, and reticularis. If you want to have a detailed session on this, you can just search in my YouTube channel. I would also like to add the development of pituitary gland. Again, pituitary gland is derived from two different sources. The first one is the ectodermal pouch. Here you have the oral cavity or the pharynx, and from the pharynx you have a diverticulum forming, and that is known as the Rathke's pouch, or this oral ectoderm is actually giving rise to Rathke's pouch once it gets pinched off from the oral cavity. Likewise, you have the diencephalon, the neural epithelium, which is giving rise to a diverticulum, or you call it as infundibulum. So now you have to know what are the derivatives of each. Rathke's pouch, which is derived from the oral ectoderm, and the infundibulum, which is derived from the diencephalon. What are the derivatives? So Rathke's pouch, if you have a closer look, you can see that there is an anterior wall, a posterior wall, and there is a cleft in between. So the anterior wall is actually giving rise to pars anterior. The posterior wall is actually giving rise to pars intermedia. It's not pars posterior. We usually tend to say pars posterior. No, it is pars intermedia. And some of these cells will actually encroach onto the infundibular region to form pars tuberalis. Now, coming to the infundibulum, the derivatives of the infundibulum are, it will first form the stalk and the remaining portion will be forming the pars nervosa. So these are the different parts of the pituitary formed from two different sources. So suprarenal gland and pituitary gland, since they are having two different sources, again, these will be we know that in the forebrain, on either side of the forebrain, in course of development, a dipping will be formed. This is known as optic sulcus. Okay. Then what happens is this sulcus will get deepened and it will finally form two important structures. This is known as the optic stalk and the distal dilated portion is known as optic vesicle. So the optic stalk and optic vesicle are actually prolongation of the forebrain. Now what happens is this will reach the ectoderm because the brain vesicle will be lying just below the ectoderm, right? So the point where this optic vesicle reaches the ectoderm, at that region, the ectoderm will be actually modified to form a placard known as optic placard. So that will be forming the optic placard, otherwise known as lens placard. Now this optic placard or lens placard will start invaginating. You can see that it was a flattened structure here. Now what happens is this will start invaginating. So when it starts invaginating, the optic vesicle cannot stay like that, isn't it? So it also invaginates so that it will accommodate this developing lens vesicle. So the lens placard which is formed here will invaginate and it will ultimately get separated from the ectoderm to form a vesicle that is known as lens vesicle. So you have two vesicles here. Optic vesicle formed from the brain and the lens vesicle formed from the ectoderm. So in order to accommodate this lens vesicle, what happens to the optic cup? It will also invaginate. And now you can see that the optic cup is having two layers, the outer layer as well as the inner layer. And the space between these two layers, you call it as intraretinal space. Now, as the optic cup invaginates, this will actually give rise to a fissure. Can you see a fissure? This is actually getting invaginated, but they are not actually meeting. So there is a fissure formed within the optic cup and along the optic stalk. So this is known as choroid fissure, and the purpose of the choroid fissure is for the transmit of hyaloid vessels, the hyaloid artery and hyaloid vein. And we know that in course of time, the hyaloid vessels will just degenerate and the proximal portion will be actually forming the central retinal vessels. So hyaloid vessels will be first supplying the developing lens, and you know that after birth the lens is an vascular structure. So what happens, the distal part of the hyaloid vessels will just degenerate and the proximal portion will be forming the central retinal vessels. So something about the formation of lens in detail. We know that this is the optic placard or lens placard. It will get invaginated and it will form a lens vesicle and it will get separated from the ectoderm. Now the lens vesicle is actually lined by somewhat cuboidal cells. So it has got an anterior wall as well as a posterior wall. Now what happens is the posterior wall will elongate. The cells of the posterior wall will elongate and they will actually lose their nuclei to form the primary lens fibers. So the posterior wall cells lose their nuclei to form the primary lens fibers, and they will actually reach the anterior wall, and this space, this cleft between the anterior and posterior wall will get obliterated. Further addition of the lens fibers, the further addition you call it as secondary lens fibers. They are actually added from the cells at the equatorial region. Now, at this point, you know that we have the hyaloid vessels which are supplying the lens, and they will be actually forming due to the blood supply. There will be a vascular membrane covering the lens. But soon after the degeneration of the hyaloid vessels, this vascular membrane will just disappear and ultimately the lens will become avascular. So why I mentioned about this vascular membrane is there is a pupillary membrane which is lying anterior to the lens vesicle, and that is actually covering the lens for a period of time, and later, say after 7 months, it will just disappear so that we get a definite pupil. Now, something about the formation of retina. We know that this is the optic cup. By the formation of lens vesicle, this is actually becoming bilayered. You call the two layers as the outer layer as well as the inner layer. The outer layer is actually giving rise to the formation of retinal pigmented epithelium, and the inner layer, you can see a pink color here and a yellow color here. So the inner layer is actually forming two parts, anterior smaller portion and a posterior larger portion. So the posterior larger portion is actually forming the sensory layer of retina or the retina proper, and the anterior smaller portion will be giving rise to ciliary body and iris. Now something about the formation of vitreous body. So vitreous body is actually developed in two stages. First we will have the primary vitreous body developed, then we have the definitive vitreous body. So these two bodies are having two different sources. Primary vitreous body is ectodermal origin. The cells between the lens vesicle and the optic cup, which are ectodermal, they will be actually prolonging to form the primary vitreous body. Later what happens is the mesoderm, which is lining the optic cup, that will be actually growing into the primary vitreous body to form the definitive vitreous body. And the process from the vitreous body, the definitive vitreous body, will give rise to three important structures. One is the suspensory ligament of lens, then you have the hyaloid membrane and the hyaloid canal. All these are actually derived from the definitive vitreous body. So when you're asked about the formation of vitreous body, we have the primary as well as the definitive one. Primary is ectodermal in origin, whereas definitive is mesodermal in origin. You have to keep that in mind. So I mentioned about the formation of lens, mentioned about the blood supply of the lens and the formation of a vascular membrane covering the lens. So anteriorly, sometimes that vascular membrane won't go off, and this is the clinical condition. So can we have the poll? Identify the clinical condition. That vascular membrane is not actually disappearing completely. So what do you call this clinical condition? Very good. That is known as persistent pupillary membrane. So in the beginning, we have the pupillary membrane, but later on what happens is the pupillary membrane will just gets degenerated once the anterior choroidal vessels, which are supplying the anterior aspect, gets degenerated. But if it is persisting, it will be seen as a membrane in front of the lens. And that is known as persistent pupillary membrane. Now, let's move on. So that's the end of today's session, the systemic embryology under the topic central nervous system and development of eye. Can we have some questions from this session, Priyanka?
Thank you, Dr. Rose. Thank you for helping all the participants revise these crucial topics in embryology. We have received a lot of questions from the participants here. So we have picked up some questions that you can answer right now.
Okay.
The first question, Dr. Rose, is, can you please tell what all to write when asked about neural tube defects?
Okay, so neural tube defect is a very favorite question for most of the examiners, and this occurs due to a defect in the neurulation process, say roughly during 3 to 4 weeks. And it can be, this defect can be either open to the surface or it can be closed with the skin. Now, open neural tube defects are more severe, and we have already seen a condition known as craniorachitis, where the entire length of the neural tube is actually opening to the surface. And we can also say about the different types of neural tube defect. When the anterior neuropore is not closing properly, we call it as anencephaly and the associated anomalies. And if the caudal neuropore is not closing properly, we can say that condition as spina bifida and its different grades. Then neural tube defects are multifactorial. That means it can occur due to genetic or environmental factors. And the role of retinoic acid, insulin, and high plasma glucose in the formation of neural tube defects have already been proved. And the folic acid supplementation can actually reduce the incidence by 75%. So all these points you can include when you're asked about the neural tube defects. Any more questions?
Thank you, Dr. Rose. So one more question we want to take up now. This question is, has plane of retinal detachment got any embryological explanation?
Actually, we have already seen that the retina is made up of two layers, embryonically speaking, isn't it? The outer pigmented layer and the inner neuronal layer, or the sensory retina. So there is a potential space between the pigmented layer and the neural layer, and that is known as intraretinal space. So in the adult, the sensory retina is so closely linked with the retinal pigmented epithelium that the production of visual pigment relies on this juxtaposition. So in the human body, wherever there is a potential space, if there are favorable conditions, what happens is the fluid will get automatically accumulated in that potential space. So there will be fluid collection in this intraretinal space, and the two layers, the outer pigmented layer, will get separated from the sensory layer. And what happens is the retina can no longer function and the sight is lost in the detached portion. So retinal detachment, I think it is actually a misnomer, because retina is not getting detached from anywhere. It is just the splitting up of the two layers of retina in the space which was there embryologically. That is the intraretinal space between these two layers. If you get a fluid collected, the pigmented layer can no longer lie closer to the sensory epithelium. So the retinal detachment, the retina is not actually getting detached from anywhere. It is just the filling of the intraretinal space which you have during the embryonic period.
Thank you, Dr. Rose. So we will end the Q&A session here, but just to reiterate, because we have a lot of other questions that are still remaining. So we will send the answers to all those questions along with the questions of the previous two sessions later to the participants on email. And also a recorded version will be shared with the participants for all the three sessions. Dr. Rose, any closing remarks from your end?
So I'm happy to see many of you attending these three sessions. So this was just meant to be a revision session. I know it's not a detailed session, so many of you might have found it difficult and many of you might have found it easy. So it is just a quick wrap of all the topics which we usually come across for the exams. So if you want a detailed session of all these topics, you can very well go to my YouTube channel. And all the best for the exams. And thank you, Xaviour team, for giving me this wonderful opportunity.
Hello, ma'am. This is Kritika here. There are a lot of students who are asking. There are certain questions, if you can recommend or suggest, which usually comes in the university. I guess students are really worried about the exams coming up. So we can just mention 2 or 3 questions related to this particular topic, which they should be focusing on, or something which comes in the universities, which can help them.
Actually, from the central development of central nervous system, the neurulation is a favorite topic. Neural tube defects, neurulation. Then in the previous sessions, I've already shared many questions. Then anencephaly, spina bifida. You can just go through the different grades of spina bifida. Then development of eye, development of lens. Development of lens is again separately asked. Then development of retina. All these are favorite questions as far as this session is concerned. The previous sessions, I think I have shared most of the questions which can be asked.
Great.
And of
Thank
course,
you,
the brain vesicles. We have already dealt in detail about the 3 brain vesicles: prosencephalon, mesencephalon, rhomencephalon, and their derivatives. That's it.
Thank you, Dr. Rose. And I would like to thank everyone who attended the masterclass session today. Request you all to post your feedback about this session in the Q&A section, and we'll be able to follow up then. Please keep connected to the Facebook page of Xaviour India for further updates. Thank you.